All right, let's get started. Good morning, everyone. My name is Kathy Grunling. I am the co-lead for the Georgia CTSA Collaborative and the Multidisciplinary Team Science function. Part of what we do is try to encourage collaboration across all of our institutions and others working in science in the Atlanta area. So today we're going to do a speed networking event on cognitive neuroscience. These are fun, they're fast. We hope that you will meet others who you'd like to collaborate with. I'm going to just talk a little bit about some ground rules. You want to advance the slides, Lauren? Thanks. So during the presentations, everyone will be muted except the presenter. Each presenter is going to have 4 minutes. Lauren will be advancing the slides for you. It's the most efficient way to do these quick presentations. There'll be a small on-screen timer, so you'll be able to see where you are in your 4 minutes. At 4 minutes, if you're still presenting, we're going to gong you off the stage. It'll be a very loud gong noise. You'll have a few more seconds to wrap up before you get muted. So the important thing to know here is that our presentations are random order, so you won't know when your presentation is scheduled, so stay on your toes. Next slide. So while this is going on, we don't have a lot of time between presenters to ask questions. So what we'd like you to do is use the chat feature to start a conversation with someone who you'd like to follow up with. We'll have some time at the end for a more general discussion, at which point you can raise your hand and we'll call on you to unmute yourself. If you're not sure where the raise your hand button, it's under the participants button at the bottom of the screen. You can also find it under React. Lauren will provide you with the presenter book, which contains the contact information for everyone who's presenting. So if you want to follow up with someone afterwards, you'll have their contact information. So that link, we'll put that link in the chat shortly, and we'll send the book in a reminder email, and it's also in the calendar invite. So with that, I'm going to turn it over to our host today, who's Tansu Chiliko, who's a professor and chair in the School of Psychology at the Georgia Institute of Technology. And I'm really pleased that he's able to lead the discussion today. Tansu?
Thank you so much. First off, please join me thanking Lauren and Kathy for the initiative and Lauren for organizing the event so expertly. And thank you so much for everything that you do for the entire community. So basically, Greater Atlanta Metropolitan is an exciting region to study brain and behavior. And in particular, in cognitive neuroscience, we have a rapidly growing multidisciplinary community. And there are so many exciting developments across the region that involve creation of new centers, degree programs, increased number of students studying in the programs at the intersection of brain, cognition, and behavior, new training grants, and much more. Just to give you a few examples from Georgia Tech, as you know, just like other USG universities and Emory, there are interdisciplinary initiatives, research centers, one of which at Georgia Tech coming up this summer is called Neuroscience, Neurotechnology, and Society. And there's a new PhD program in neuroscience and neurotechnology that actually started and registering the first cohort of students who are going to start in August. There are 3 new centers of excellence that have been established, and one of them on navigation, the other one is computational cognition, and the third one is human AI robot teaming. And there are many of the speakers over here, they are member of these new centers. Each one is flourishing. They have grants and they have started organizing community events, bringing opportunities in terms of collaboration. And each center has a focus that is of interest to broader cognitive neuroscience community as well. And today's roster is actually a great indication of the breadth and depth of research that we have in cognitive neuroscience in the region. We have speakers from UGA, Morehouse, Georgia Tech, Kennesaw State, Emory, Georgia, University of Georgia, and Georgia IP Alliance, talking about a broad range of spectrum of research, ranging from brain development all the way to the neuronal control of curiosity. So after the presentations, we will have time for Q&As and start the discussions. on how we can maximize the synergies in our community to make lasting impact. But now, without further ado, I would like to actually introduce our first speaker with drum rolls, and that is Ashley Sanders. Dr. Sanders is an assistant professor in the Department of Psychology at UGA, specializing in developmental cognitive neuroscience. Her research focuses on how socioeconomic disadvantage influences children's brain development and risk for psychiatric illness. And she utilizes neuroimaging, inflammatory biomarkers to explore neuroimmune interactions and study under-examined regions like cerebellum along the process. Thank you so much for being here, Ashley, and the floor is yours.
All right. Well, thank you so much for organizing this. I'm really excited to chat about my research and hopefully get to learn more about you all and see where we all can collaborate with each other. So as I was just told, I'm a new professor or assistant professor in the Department of Psychology at the University of Georgia. I started in fall of 2024. I came from WashU in St. Louis, working under Deanna Barch and Joan Luby, where I focused in a lot on the socioeconomic impacts of brain development in children. So I'll be talking a little bit about, very briefly, my research overview, and then go into where I'm headed with this. So at WashU, I first started working a lot on the neonatal period of brain development through adolescence. So I'm carrying that over here at UGA, and we're really excited to get started. So you can go ahead and advance the slide. Okay, so here is my general overview. We are currently recruiting children in the greater Athens to Atlanta area, aged 7 to 10 years old, from a broad spectrum of disadvantage. So not just simply focusing on income, but also the range of neighborhood level and community factors, such as area deprivation, that we know define this broad spectrum of disadvantage across the lifespan. One thing that we focus a lot on in my lab is the impact of inflammatory cytokines that are circulating in the blood on brain development. We know that inflammation has a significant impact on specifically white matter, which is one of my main modalities of diffusion imaging that we use. And so we want to see how these different socioeconomic or disadvantage variables differentiate that relationship. And the overarching question that we're asking, though, is what are the biological mechanisms by which early life disadvantage impacts how children really perceive and interact with the world around them? So we can go ahead and go to the next slide. So as was alluded to, I am really focusing on the cerebellum as a novel target in disadvantage. So through a combination of genetic blueprints and environmental information, we know that an organism grows in a manner that allows adaptation to and successful interaction with the setting in which they develop to ensure survival. And a lot of these adaptations in disadvantaged settings may impact how children process information about their environment. But more specifically, this sort of predictive coding framework that is largely housed in the cerebellum, but then is projected to the cortex, it builds an internal model of the external world, and it's constantly transmitting these prediction errors to and from the cerebral cortex. But this alters how a child then processes environmental stimuli and predicts consequences to subsequent interactions with these stimuli. So because the cerebellum has a really prolonged maturation and postnatal neurogenesis, it is uniquely impacted by the impacts of inflammation, which we're proxying as far as the circulating cytokines in the blood. So we're really interested to see if these environmental pressures differentially alter the cerebellum's developmental trajectory and thereby impacting brain-wide mechanisms. Okay, you can go ahead and advance to the next slide. So what I can offer to collaborators is specifics on neuroimaging analytics. That is one of my specialties, specifically diffusion and resting state functional connectivity, and expertise in disadvantage-related development from the neonatal period through adolescence. But what I really need help with is reaching out into more rural communities and community outreach and engagement in the Athens to Atlanta area. And also, if anyone has any further knowledge about tasks that we can employ for resting state, or I'm sorry, task-based functional connectivity for predictive coding mechanisms, And I have some tasks that we're developing right now, but not enough time to talk about them. I'd love to talk offline, but yeah, that's where we are going. So thank you.
Perfect. Thank you so much.
Thank you.
So our next speaker is Dobi Rahnev. Dr. Rahnev is an associate professor and associate chair of research in the School of Psychology at Georgia Tech, where he leads the Computational Cognitive Neuroscience Lab. His research delves into computational and neural mechanisms underlying perceptual decision-making and visual metacognition, employing techniques such as functional MRI, transcranial magnetic stimulation, and computational modeling to explore how the brain processes sensory information and confidence judgments. Dobby loves soccer and plays every week. Knowing Dobby that he does everything to perfection, I won't be surprised if we see him playing for the national team sometime soon. The floor is yours, Dobby.
Thank you, Tansu. All right, so as Tansu mentioned, I'm an associate professor in the School of Psychology, and I focus on metacognition and perceptual decision making, as well as cognitive modeling and artificial neural networks. So if you go to the next slide, I'll expand on that a little bit more. So in terms of domains, there's two main domains of focus for my lab's research. The first one of them is metacognition. And as you know, metacognition is the ability to introspect on our own cognition. Most of the time in the lab, we study that using confidence ratings, but it doesn't have to be just confidence ratings. It can be other ways of introspection. So my lab really specializes in computational models of metacognition, as well as measuring metacognitive ability in a formal and quantitative manner. We're probably one of the top few labs in the world who do those two specific things: computational models of metacognition and measuring metacognitive ability. Beyond that, we have developed a lot of paradigms within perception mostly, but not only, that produce confidence actual dissociations. So this means that we have developed conditions that are sort of similar to each other, but and people are equally accurate in both conditions, but in one of the conditions they're more confident. So that allows us to really uncover interesting mechanisms. Within this domain, I have led a few different initiatives. I developed the confidence database. This is the largest database of open data related to confidence ratings. And recently I organized an effort to decide on consensus goals for the whole field of visual metacognition. So that's one. The other one is perceptual decision making, where we study how people make decisions about noisy and ambiguous visual stimuli. This involves computational models again. We've developed a lot of experimental paradigms. We studied the neurocorrelates. And as one example, a few years back, a collaborator and I published a paper on suboptimality in perceptual decision making that was in the Behavioral and Brain Sciences journal. So it came with 27 commentaries and all of that. So if you go to the next slide, I can tell you about our techniques. So the main technique by far is cognitive modeling. So things like signal detection theory and the drift diffusion model is something that is the bread and butter of the lab. So if any of you ever need help with that, definitely very happy to collaborate. We also build a lot of custom computational models of this type. Beyond that, we work with artificial neural networks. So we have developed our own neural networks, but also use existing ones, mostly in the go to try to understand to what extent they differ, to what extent they're similar to humans, and help us infer underlying principles and mechanisms. In terms of cognitive neuroscience, we use TMS fMRI as well as concurrent TMS fMRI. So probably more the TMS part is relevant. So if if you're interested in TMS and you can do it, maybe we can collaborate as well. So if you go to the next slide, to wrap up, what I can offer collaborators is computational modeling expertise, so cognitive models, artificial neural networks, as well as domain knowledge in metacognition and perceptual decision making. What I need help with, or what we can really benefit from, is access to special populations, especially clinical populations, but any kind of, non just college undergraduates, which is our bread and butter. Any kind of data set that include confidence ratings. So we mostly study within perception, but we can expand to other things, as well as just metacognition more broadly. So if you're interested in that, please talk to me. Thank you.
Thank you, Dobby. We are going to applause all the speakers at the end, but in between, please do feel free to send digital applause, thumbs up, and love signs. But we are going to continue now next with Kaushar Ulia Chatnuri. I'm sure that I'm pronouncing wrong. My apologies. From Kennesaw State, I'm going to be talking about prenatal cannabinoid exposure alters memory by modulating glutamatergic neurotransmission. The floor is yours.
Hi, thank you, Dr. Tanzu, for the introduction. My name is Kaustubh Chaudhary, as introduced, and I'm representing Dr. Vishnu Subramanian's lab. And one of our collaborator, Dr. Erika Holiday, she will also represent some of her work after a while. So, and the focus of our research is molecular mechanisms of learning and memory. And as you can see, the title of our research is Prenatal Cannabinoid Exposure Alters Memory by Modulating Glutamatergic Neurotransmission. Now, the question may come, why did you choose cannabis? So cannabis is one of the most commonly used illicit substance, and as of now, 24 states in the USA has legalized the use of cannabis, and with time it's increasing. And it was found that the pregnant women who takes cannabis during pregnancy, their children shows difficulty in learning and memory during adolescence, but the exact mechanism is not known. Next, please. Next, 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 next. Okay, so if anything happens, any alteration happens in the brain, the first thing that will show up to us is the behavior. And you can see the center of the figure, I'm showing the behavior. And each behavior represents a certain function of a certain brain areas. So that means alteration in behavior means alteration in certain brain areas. In our case, we are dealing with spatial memory. And so we are talking about hippocampal alteration. As brains are composed of neurons, billions of neurons, regional or the hippocampal alteration means something is going wrong at the neuronal network level, which eventually is the result of cellular malfunction. And if we look at the center or at the deep root level, that means something is going wrong at the synaptic receptor level. Maybe the receptor expression is altered or the function is altered. So what do we do to find out all these mechanisms? Next, please. Next, 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 next. So you can see for each level, we have different kind of methods that we use in our lab here at Kennesaw. Like for the behavior, we do different behavioral tests like open field test, contextual fair conditioning, and Morris Watermaze, and some others. At the regional level, we record the activity of hippocampal regional function by long-term potentiation or long-term depression recording, which is a cellular model of learning and memory. And then at the network level, we record microelectrode array to record the glutamate release or glutamate reuptake. At the cellular level, we record the activity of individual neuronal cell. And at the synaptic level, this is interesting because it's not possible to go at the synapse because it's so small in size, angstrom in size. So my advisor, Dr. Vishnu Subramanian, has developed a unique technique through which we can isolate synaptosome or synaptic contents and record the activity of individual ion channel. And in addition, also, obviously, Western blot, everyone knows we can record the, see the expression of the receptors. Next, please. Next, 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 next. So did you find any mechanisms or changes? Yes. At all the levels that I discussed earlier, we found some changes, like for the fear conditioning or for the LTP recording, we saw alteration in the plasticity, or at the cellular recording, as well as at the single channel recording. All of them shows some kind of changes, which says something is going on. Next, please. Next. So to wrap up, what we can collaborate or what we can offer the unique technique in addition to all the others one that I said, by which we can record the activity of individual ion channel. And what we need help with, if we can have something to measure the epigenetics modification or electrophysiology of free moving animals, optogenetics, that will be great. Thank you, everyone.
Thank you. Perfect timing. Now, the next talk is going to be by Vincent Costa. Dr. Costa is an associate professor in the Department of Psychiatry and Behavioral Sciences at MRI Medicine, where he leads research focused on the computational and systems neuroscience of motivational neuronal circuits. His work aims to elucidate how these circuits influence emotion, cognition, and behavior, contributing to our understanding of the neuronal base of motivation and its role in mental health disorders. Thanks for being here, Vincent, and the floor is yours.
You're on mute.
My lab's primarily a primate neurophysiology lab. And the broad question we're trying to do is combine systems neuroscience techniques with molecular neuroscience tools to sort of profile cognitive phenotypes that we think are relevant in psychiatry in non-human primates to develop non-human primate models of these diseases. And what we've been focusing on in the lab most of late is trying to understand how amygdala circuits essentially regulate curiosity in the context of explore-exploit decision-making. Can you hit next? And this is going towards a longer-term goal of trying to understand how, if you perturb a particular circuit that you think is relevant within a broader network, how you change information processing in that network, because it's increasingly understood that in the context of psychiatry, where the dysfunction lies is in at the level of circuits and networks and not, ion channels and molecules. Next. So we are trying to combine essentially 4 domains within the lab. The first is valence-dependent reinforcement learning. So hit next. To try to make our tasks that we study in macaques as relevant as possible to human cognitive neuroscience, we've moved to using symbolic reinforcers. So we have the monkeys essentially gambling for tokens in the lab. And what this has allowed us to really do is open up the study of both repetitive and reversive learning ethically and responsibly in the context of reinforcement learning. So we can do things now like record from the amygdala and actually understand both repetitive and reversive processing in the same way that you can study these processes in rodents using primary reinforcers. Next. And then the other sort of main innovation that we use a lot in the lab is we've sort of pioneered the use of pathway-specific chemogenetic tools to perturb amygdala circuits, in this case to the amygdala inputs and nucleus accumbens. And what's been really exciting is that we've been able to take high-level cognitive tasks that people deploy in human cognitive neuroscience all the time, like Bob Wilson's, who's in psychology at Georgia Tech, horizon task, and we've been able to have monkeys complete the same task. And now we've been perturbing these circuits, and we're getting behavioral effects that align with some of the neuromodulatory effects that people have seen using TMS and TDCS in the same task. And so the fact that we can get down to the level of individual circuits in terms of phenotyping these much broader cognitive behaviors has also led to these other 2 domains. So hit next. In the lab, we're trying to really push on how far we can link molecular neuroscience up to cognitive neuroscience. So we've been doing a lot of transcriptomic profiling using single nucleotide RNA-seq to identify the neurons that we're perturbing in the amygdala, and we've found gross differences now between the basal and lateral nucleus, and we're really excited trying to follow this up now to see what is the functional differences between these transcriptomic profiles. And the other thing we've been doing, hit next, is we've also pioneered the use of monosynaptic rabies tracing so that we can determine all of the presynaptic inputs into these amygdala neurons that we're perturbing with the chemogenetics and recording in the context of our neurophysiology studies to try to understand what's the information flow throughout this whole network. So we're starting to put all this stuff together, and in terms of what we're looking for is we need collaborators to really, we have these really rich neurophysiology and behavioral data sets, and we'd like to move more towards using AI to explore some of these techniques. and what they mean at the systems level. And the other thing is we also have a lot of resting state fMRI data to try to validate some of these tools using traditional cognitive neuroscience, and we need a lot of help in that domain as well. So thanks.
Thank you so much. Insight into brain and behavior and so many opportunities for collaboration. We are going to get back to that at the end of the session. But next, we have Amrita Harikumar from Georgia State University. And Amrita is a PhD student in the Cognitive and Affective Neuroscience lab, led by Dr. Calhoun, and collaborates with Friends faculty broadly. And her desire is to understand the neuronal underpinnings of schizophrenia using an interdisciplinary and transdiagnostic approach. Amrita loves to travel, write, and have a journal about her life from the age of 10. Well, the floor is yours, Amrita.
Thank you so much for that wonderful introduction. Welcome, everyone. I'm recovering from a cold, so I will try to project my voice as best as I can. But today, I want to talk a little bit about a project that I'm focusing on for my dissertation entitled Classifying Schizophrenic Patients and Healthy Individuals, Whole Brain Spec Functional Connectivity Using Support Vector Machine Classification. Next slide, please. So a little bit about myself. I'm a fourth-year doctoral student, soon to be candidate. I expect to graduate next May. I'm a Brains and Behavior Fellow. I'm in the Department of Psychology at Georgia State in the Cognitive and Affective Neuroscience track. My primary research focuses are using schizophrenia, functional magnetic resonance imaging, or fMRI. I've recently delved into some SPECT imaging work, which is what I'll talk about in the next slide. And I'd love to meet others to do more translational neuroimaging research, both in the schizophrenia domain or broadly, anyone using fMRI or SPECT imaging modalities. So I'll get to what I'd love to get out of with this talk and the collaborators I'd like to meet. Next slide. So a little bit about a project that I'm working on currently. We know that functional magnetic resonance imaging, or fMRI, is a powerful modality to understand functional connections in the brain. We know that synchronous connectivity, or functional connectivity, that is connections between two brain regions synchronously, is how you measure functional connectivity, and that you can also use it as a potential biomarker. And in terms of the context of understanding schizophrenia, there currently isn't a cure for schizophrenia, but we have potential biomarkers in brain imaging that a lot of groups, including our group, is trying to investigate. So a biomarker can be genes, blood tests, any brain imaging methodology that indicates a particular disorder or symptoms. So on the top right-hand side, we have an fMRI template with 7 brain networks that has been used as a potential neural biomarker called NeuroMark. And that's it's validated off of 2 large-scale brain imaging human data sets. So our goal for this research study was to apply the NeuroMark template, and it's using something called ICA, or Independent Components Analysis. That's what Dr. Calhoun primarily uses in our lab. We try to apply it to different data sets with schizophrenia, with depression, bipolar disorder, Alzheimer's, autism, you name it. You can use ICA for different things. But we saw that there was a gap in the literature, that this hasn't really been applied to SPECT data. And the reason that we were so interested in applying this to SPECT data is SPECT is also a measure of blood oxygen level dependent flow, or bold flow. And so because it's so similar to fMRI, we wanted to see if we applied ICA to SPECT data, and combined it with a neuroimaging classifier such as support vector machine learning, if it could well classify some of the brain networks that we've seen in fMRI studies in schizophrenia. And so previous fMRI studies have shown that individuals with schizophrenia suffer from auditory hallucination delusions, and we ran SVM on the SPECT data. What we saw was that the auditory, sensory motor, and subcortical networks were validated up to 83% classification accuracy. And so ICA can be a potential biomarker for accurately classifying patients and improving symptom diagnoses. Next slide. So at the Trend Center, what I can offer is connections and collaborations with the Trend Center. That's currently where I work. I also have 10 years of experience in academic research, clinical research experience working with diverse patient populations. What I would like to do is eventually move into the industry space. So I'd love to have translational collaborations and industry networking opportunities and contacts within the Atlanta area. Next slide. I don't know if that was it. Oh, never mind. Go back. Sorry. So yes, I would like to offer my services in this area. Thank you.
Wow. Another perfect timing. Thank you so much. All right. Now, talk number 6. It's going to be by Anna Abraham from the University of Georgia. Dr. Abraham is the Paul Torrance Professor of Creativity and Gifted Education at UGA, where she also directs the Torrance Center for Creativity and Talent Development and leads the Creativity and Imagination Lab. A psychologist and a neuroscientist, her research explores the neurocognitive mechanisms underlying creativity and imagination, including topics such as mental time travel, self-referential thinking, and reality fiction distinction. Thank you so much for being here, and we look forward to your talk.
Thank you so much. Hello, everyone. As he said, my name is Anna Abraham. I'm from India. My training is in psychology and neuroscience. Did my undergraduate in India, my postgraduate degree in psychology in England, and my PhD in neuroscience in Germany. I see myself as a multidisciplinary and a cross-cultural scholar. Next. I currently, as of 2020, actually 2021 is when I arrived at UGA. I've been at UGA. This is my first kind of appointment in the US. And in UGA, I kind of don a couple of different hats. So I'm mainly based in the College of Education, where I serve as the Paul Torrance Professor in the Department of Educational Psychology. We have a range of different foci there, from gifted and creative education, applied cognition development, quantitative methodology, and school psychology. I also serve as the director of the Torrance Center for Creativity. We run lots of different types of training programs for teachers, educational professionals in general, that are centered around creativity. We also offer camps to our local community here, summer camps that are centered on creativity for young children. I also serve as a professor in the neuroscience program and the integrated life sciences program at UGA. I'm also a faculty fellow at the Institute of Artificial Intelligence, so my collaborations extend there. Next. My areas of expertise are the following. My primary area has been in creativity across in multiple lenses that I'll get into in a second. But I also focus on imagination more widely. I look at the reality fiction distinction. So how can we tell that something is real compared to not real? Mental time travel, how do we project ourselves to think about our past and our futures? Mental state reasoning, how is our reasoning about how other people think and behave similar to the way we think about our own behaviors and cognitions? Self-referential thinking, social cognition, and increasingly more now I'm developing a sort of neurophilosophical paradigms, and I also focus on aesthetic experience. Next. My works include the following books. A lot of my work has been empirical papers, but I try to consolidate what I do into book form. So these are the 3 most recent things that I've worked on, long form things that I've worked on. Next, please. My interests are very, very broad. In the neuroscience of creativity, very broadly. The kind of studies I've done involve fMRI, EEG, ERP analysis, and neuropsychology, so study of clinical populations in general. I focus on the psychology of creativity very broadly, mainly cognitive, but also elsewhere. I look at development across the lifespan, look at personality, motivational variables, and so on. I also engage in the cross-disciplinary study of the imagination, looking at aesthetics, imagery, intentionality, altered states like dreaming and so on. And I also do a lot of theoretical work in the field of creativity and imagination. Next, please. So in terms of what I can offer to collaborators next, I think anyone who's interested in kind of cross-disciplinary expertise in relation to creativity, imagination, and sort of related topics of study, such as openness to experience, play, flow, problem-solving, intuition, insight, and so consciousness, and so on. I could offer those sorts of expertise to people who are interested. Next. Expertise in translational research, from implementing insights from basic research to applied domains. The kind of domains I've worked on include public health and education. Next. And I can also offer kind of, for those who are interested, multi-level approach to research scholarship, which goes from a body of empirical studies to methods papers, theoretical frameworks, and applied perspectives, and sort of rinse and repeat the whole time long. What I need help with. Next, I'm looking for access to clinical populations for neuropsychological studies, either neurological or psychiatric. This is where my work has kind of dipped off a little bit since I've moved here. Next. I'm looking for people who are interested in sort of cross-center working groups or consortiums on creativity and innovation across a range of contexts, such as in the age of AI. And next. And finally, collaborators who are open to examining big questions and adopting cross-disciplinary approaches on topics that are relevant to creativity and imagination. So more of a theoretical bend, if you see what I mean. So theoretical neuroscientists out there. Thank you so much.
Thank you so much. Up next is Scott Moffat from Georgia Tech. Dr. Moffat is a professor of psychology and the associate chair for graduate studies in the School of Psychology. His research focuses on how aging and hormonal factors such as cortisol and testosterone affect spatial cognition and memory. He uses neuroimaging and virtual reality techniques to explore the neuronal systems underlying navigation and cognitive decline in older adults. The floor is yours, Scott. I believe you are muted.
My apologies. Thanks for coming and good morning to everybody. So my research takes place in two domains of interest. So the first one is on spatial cognition, and especially how spatial cognition changes in the context of human aging. And I'm especially interested in studying spatial navigation in particular, how younger and older people learn their way through new environments, the kinds of cues that they use, and then the context of aging, how those brain mechanisms can change with age as well. So I do this works using virtual environments, structural MRI, functional MRI, and other behavioral and brain imaging methods as well. And the second area of interest that I have is on biomarkers and modifiers of cognitive and brain aging, especially in the context of endocrine changes in the body. So I'm specifically interested in how gonadal or sex steroids might be neuroprotective agents in the context of aging, how adrenal steroids like cortisol might be serve as risk factors for brain and cognitive aging. And then my most recent interests are in glucose and insulin metabolism and how that can affect the aging brain. Next, please. One uniting factor in these two domains is my interest in individual differences. So in the context of aging, especially, some older people show a lot of age-related cognitive change, and some show little to no change at all. So three figures illustrate this. The scatter plot on the left there just shows some navigation error increase in the context of aging. Aging is on the x-axis and navigation error is on the y-axis. So you can see a general, increase in errors with age. But if you look at the bottom right quadrant, you can see there's a lot of older people in their 70s and 80s who perform as well as younger people in their 30s and 40s. And then if we skip over to the right scatter plot, it's actually what's called a spaghetti plot. This is a way to present longitudinal data on an individual basis. So each one of those little lines is a rate of change of an individual person in hippocampal volume over time. So you can see there's huge individual differences just as the lines are sort of represented up and down across the y-axis, but more importantly, the individual differences in the slopes of the lines with age. So why these individual differences? Of course, there's a whole host of reasons. I'm especially interested, next please, in hormonal and metabolic factors. So as I noted a second ago, I'm interested in gonadal steroids, especially testosterone in men as a possible neuroprotective factor, cortisol and glucose and insulin regulation as possible risk factors. Next, please. So I've done studies linking gonadal steroids, especially testosterone in men, as a protective agent in Alzheimer's disease, and the stress steroid hormone cortisol as a risk factor for Alzheimer's disease. And next, please. Next. Most recently, I've been interested in glucose and insulin regulation and dysregulation. Those plots right there show you how people with prediabetes and normal people respond to glucose ingestion. So you can see that very sharp rise in glucose and insulin in people with prediabetes, but also vast individual differences. And then next and next and next. Okay, and then we link, sorry, one less next. We link those responses in glucose and insulin in an oral glucose tolerance test to functional MRI outcomes and cognitive outcomes, and you can see some of the results from those studies right there. In terms of my interests, I would love to work with some people who have some knowledge of inflammatory markers that might serve as mediators between these hormones and brain and cognitive changes. Thanks.
Thank you so much, Scott. We all know who's next. That is Erica Holday. She's an associate professor of psychological science at Kennesaw State. She's the founder of the Neurobiological Examination of Addiction, Recovery, and Related Disorders. I suspect this is the Nerd Lab, where she investigates the neurobiological and psychosocial mechanisms underlying substance use disorders and recovery processes, focusing on factors such as adolescent development, perinatal exposure, and maternal care. Erica owns and operates a construction company on the site with her husband and will start the Bachelor in Construction Management degree in 2026. Well, we admire your ability to juggle multiple things, Erica, and looking forward to your talk.
Got to get that second career going with the current administration. It's fine. Also, I'm an assistant professor. I go up for tenure this summer, and I have what's known as a squiggly line career because I did rodent research for pre-doctoral and my postdoc, and then transitioned into human work to work with the Collegiate Recovery Center at Kennesaw because I had lost over a dozen friends to the opioid epidemic, and I wanted to make more impact with my research. And then 3 years into that, we got a vivarium at Kennesaw, and then they recruited me to start doing the work there. So I work with Dr. Kalsar and Dr. Vishnu with their behavior and molecular mechanisms of learning and memory. But I also do human work, so truly translational in nature. And I do focus on the underpinnings of addiction with the influence of early life adversity. Next slide. I will also say my models are not nearly as sophisticated because I jumped into human work in 2020 when I was hired at Kennesaw. So I'm definitely looking for senior collaborators. On the clinical side, I work with all undergraduate researchers, and we're looking at single nucleotide polymorphisms using qPCR. And we have our participants do a battery of surveys, and then they do some executive functioning tasks. And what we're finding there is the MET alleles for both BDNF and COMT are impairing cognitive flexibility while sparing other domains like working memory and impulsivity measures. And then the preclinical models, I'm using a limited bedding and nesting model to mimic adverse maternal care and looking at outcomes in both the dams or the mothers and the pups. And we combine it with substance use during pregnancy or gestation using THC right now, but we can also do nicotine, alcohol, opiates, or medication-assisted therapies. So that allows us to do some more mechanistic studies. Next slide. So these are some of the results from the studies that I've done over the last 2 or 3 years. On the left is looking at a comparison between our students in the Collegiate Recovery Program and then age-matched college controls in an oddball task. And what you can see in the blue line at the top, we have an augmented P300 wave to the rare event and an oddball task, and a delayed weakening of the P500. And we've actually been able to use this data to go to our university to support having additional space and curriculum for our students that are in this program. And then on the right side of the screen, the phenotypes of stress, I was looking at microRNA expression that were extracted from exosomes and saliva, and this was recently published in Frontiers in Psychiatry. And we were able to determine that there are differences in the microRNA profiles of individuals that had high stress at the moment that it was collected and also high adverse childhood experiences. And at least one, the microRNA 135A, has a different profile for individuals that didn't have early life adversity, but were experiencing current levels of high stress. And we're looking at ways to market that for a non-invasive clinical screen. Next slide. And then to wrap it up, what I can offer to collaborators is that microRNA process or extracting from saliva, or can teach those methods. I also have collegiate recovery insight, and access to that diverse population, although it is a very difficult population to recruit from, and not just collegiate recovery, also all ages across recovery. And I am also the research coordinator in our psychology department. So I have access to our SONA participant pool, and Kennesaw does have the largest enrollment of psychology majors. So we typically have anywhere between 1,200 to 1,700 enrolled in our Psych 1101 classes. So if you're looking for a population like that, we can certainly collaborate. And then what I need help with is more senior mentoring because we don't have it at Kennesaw, especially experimental designs with SNPs or genetic markers, and also in seeking extramural funding for those models. Thank you.
Thank you so much, Erica. All right. Our next talk is going to be by Piyush Padi. Piyush is a graduate student at UGA. He's developing a novel genetically engineered microbial technology for PD and AD. And his talk is entitled Microbiome-Based Engineered Live Biotherapeutics to Treat Cognitive and Behavioral Symptoms of Alzheimer's Disease and Related Dementias. The floor is yours, Piyush.
Hi, yeah, I'm actually, thanks for the introduction. I'm actually moved on to become a postdoc now. So I'm in the Dr. Anu Kanthasami's lab, who's the director of the Isaacson Center of Neurological Disease at University of Georgia. And in our lab, we focus on multiple aspects of Parkinson's, Alzheimer's, and other neuropsychological diseases like major depressive disorder. But one arm of the lab that we focus on is actually using engineered microbiome therapeutics for neurological disorders. So, unlike the clinical studies that have been presented here, our lab is mostly focused on the preclinical aspects of this disease and understanding the molecular mechanisms. Next slide, please. So one of the things that we focus on is understanding what the gut-brain connection is in the context of neurological diseases. I'm sure all of you have been in the news, have been familiar with a lot of impact of how gut microbiome can impact neurological function. And in our lab, we kind of focus on two things in focus in Parkinson's disease specifically. One is looking at the microbiota enteric neuroimmune axis, understanding how the microbe interact with the enteric nervous systems and how that modulate the neuroimmune connections in the brain. And the other aspect that we also look at is therapeutically targeting some of these neurotoxic gut metabolites that are produced in for Parkinson's disease. For example, foods that are rich in choline and betaine, which is like usually the high-fat diet, high-meat diet, they produce these toxic metabolites called TMA, which then gets converted to TMAO, which is trimethylamine oxide. And the TMAO has been shown to cause these pro-inflammatory processes in the brain. So we wanted to understand what is the role of the gut microbiota and can we modulate the gut microbiome to understand these neurological processes. Next slide, please. The other aspect is understanding like how can we protect these pathophysiological mechanisms by targeting these metabolites by using these engineered therapeutics. The reason of using engineered therapeutics is we know that in the market there's a lot of probiotics, but we thought that by engineering them specifically to produce specific metabolites acting as a drug, we can modulate some of these neuronal and even glial functions. So in our three arms that we look at for specifically for Parkinson's disease, we have developed a system that we produce L-DOPA. And L-DOPA, we know that is a gold standard therapeutic for Parkinson's disease. So we showed that in various preclinical model systems that we are able to recover some of the classical motor phenotypes as well as some of the non-motor phenotypes that we expect. In our Alzheimer's disease project, we noticed that L-DOPA and dopamine are direct precursors to norepinephrine, which are also dysregulated in Alzheimer's in the disease context. So by giving our engineered therapeutic, we're able to also modulate some of the norepinephrine projections and thereby improve the cognitive functions. Similarly, dopamine, norepinephrine, all these play a role in major depressive disorders. So we also tested this in our animal model systems to show that we can improve some of the non-motor behavioral phenotypes as well in the depression model. Next slide, please. And so this is some of the data looking at some of the in the Alzheimer's disease and major depressive disorder. We've used our lab has a expertise in various model systems. So we've used two different types of amyloid mouse models and rat model systems for Alzheimer's disease. We've also developed some data for preclinical model. Overall, we're seeing some disease modifying effect that we observe with giving our therapeutic, and especially in the rat model systems of Alzheimer's disease. So this was kind of an exciting frontier that has not been explored yet. And in the chronic unpredictable stress model, we were able to kind of improve some of the depression phenotypes, which include the anhedonia and immobilities. Next slide, please. So what we can offer is we have a comprehensive behavioral core, a wide range of molecular techniques, access to some of the ICNDR, the Institute's core faculty, expertise in neuroinflammation and olfactory biology. What we need help with is actually enzyme design and that modulate metabolic pathway, computational modeling, and some clinical expertise specifically around depression, anxiety, and psychiatry. And we want to be able to also access some biospecimens for clinical cohorts. And with that, I'll thank you.
Thank you. Thank you so much. And my apologies for my incomplete Google search on you, and congratulations on your PhD.
Oh, thank you.
So next up is Dr. Yerat Alumi. Dr. Alumi is the founder and CEO of the TQ Intelligence and adjunct professor at the Morehouse School of Medicine. He develops AI-enabled digital mental health care products to address disparities in mental health care globally. He is enthusiastic about the promise of AI to support precision and individualized approaches to mental health treatment. Nice to see you again, Yared, and the floor is yours. I believe you might be muted. Or are you actually here?
I don't see his name on
No,
the list.
I don't. Well, then we're going to move on. Just in case he joins us a little bit later, we are going to be able to come back. So next up is, well, Dr. Apurva Ratan Murthy, or as he goes by Ratan. He's an assistant professor in the School of Psychology at Georgia Tech, where he directs the Murthy Lab focused on integrating biological vision with artificial models to understand neuronal codes and algorithms that enable human vision. His research combines advanced neuroimaging techniques, including 70 fMRI, with computational methods, and including deep neural networks and AI interpretability, to develop neuronally mechanistic models that shed light on perceptual development and disorders like agnosia. So he is an avid RC plane and drone collector and a pilot. So with these technologies and his background in creating artificial intelligence inspired by vision, we can imagine then his next generation drones and planes are going to be fully autonomous. And we look forward to your talk, Rod.
Great. Hi. Can everybody hear me okay?
Yes,
Great.
we can.
Okay. Hi, my name is Ratan. I go by Ratan. And I moved to Georgia Tech early last year. So it's been a year that I've been here. So prior to that, I actually obtained my PhD also in India, like Anna. I'm also from India. So I did my PhD at the University of Science. And after that, I did my postdoc at MIT with Jim DiCarlo and Nancy Kenwisher. And then I moved here. And now I am leading a group, and we call ourselves the Vision Cognition and Computation Group. And let me tell you about the work that we do. That's the next slide. So I've grouped it by like the three major thrusts, at least the way I think of how we're running the lab. So the first thrust really is to try to uncover the neural codes that support human perceptual systems. So for the most part, my work is centered on human vision. And our goal is really to try and come up with a simulation, so to speak, of the way human brains process visual information. To that end, we've developed, some of my prior work, we developed the first artificial neural network models of category selective regions of the human brain, which you can use for other kinds of things. For example, to figure out exactly the kinds of features that the human brain likes and so on. Then we use some of the same kinds of methods to discover a part of the brain that we think is likes images of food. So that was published a few years back. And I'm also very interested in modeling human behavior. So some work that we've done in that sphere is mostly related to visual awareness systems. The second thrust of the work that we do is, in as much as we are able to understand, how our human brains work, we want to see if we can induce the same kinds of algorithms in current AI systems. And that's really the second thrust of what my lab does. So one of the things that we did recently was to develop new AI models, new model architectures entirely, which have really modular subsystems, pretty much like the human brain does. So we call them models with brain-like topography. And we're calling these systems toponets because to distinguish it from the other kinds of categories. So we've developed vision systems, we developed language models with brain-like topography, and more recently, hopefully in the next month or so, we'll see some topographic audio systems as well. So here is one thing that we've learned from the human brains that we can just induce in AI systems. So the other thing that we've done is we come up with methods of comparing brains with models. We are calling it the Neuro AI Turing test for short. But I am actually more excited to tell you about the third thrust. Next slide, please. Which is, in as much as we try to understand neurotypical brains, there's a major thrust of our lab that really wants to understand the neural basis of atypical neurobiology. Now, in that realm, I've studied congenital blindness. We work with patients who have intracranial electrodes in their brains. And we are also working with special populations, people who have prosopagnosia. There are some people in the lab who are also interested in, along with Dobie's lab actually, in people who do not have mental imagery, for example. And we are also very interested in trying to model brain stimulation patterns. So imagine that there's a person with electrodes in their brain and you microstimulate their brain, try to recreate their perceptual experiences. Okay, so let's move on to the next slide real quick because now what can we offer? So I think our lab is really good at building models, predominantly artificial neural network based models, recurrent neural networks. The stuff that we did not talk about is that I have some work where we are trying to do like continuous RNN types. So Vincent, for example, if you're working with monkey data, that was my background. We have continuous time neural network models that we can develop. What we really need help is connections in the Atlanta Clinical Neuroscience Network, intracranial, special populations, and so on. So if anything here sounds exciting, please reach out. It's been great to be part of this community. Thank you.
Thank you, Ratan. We are almost there, last three talks. The next one is going to be by Dr. Ronghu Wu, who's a professor in the School of Chemistry and Biochemistry at Georgia Tech, where he leads the Wu research group, which is focused on advancing mass spectroscopy-based proteomics. His research specializes in developing innovative analytical methods to study protein post-translational modifications, particularly glucosylation and phosphorylation, with applications in understanding disease mechanisms and identifying potential biomarkers. Thanks for being here, Ronghu, and the floor is yours.
Thank you, Tansu, for your introduction. And also thank Laura and Casey for organizing this nice event. I'm Ronghu. I'm in the chemistry and biochemistry department from Georgia Tech. So I came here more than 10 years ago. Our research is focused on mass spec-based proteomics. Actually, we try to use mass spec to sequence and also quantify every protein in a biological sample, even though sometimes it may not be possible. We are especially interested in protein modifications and also protein dynamics. Next. Here is a list of our major research topics. We have been working on protein glycosylation. We have developed chemical and also enzymatic methods to globally and also science-specifically analyze protein glycosylation. When we talk about protein glycosylation, it plays an extremely important role in nearly every extracellular activity. Therefore, we are also interested in glycoproteins only located on cell surface and also in the cytokine. Even in the cytokine, pretty much all the proteins, they're also glycosylated. Regarding glycoproteins, not only proteins, but also the glycans contains much valuable information. Therefore, we are also interested in glycoproteins with a particular and also important glycans, such as like TN, STN antigens. Next. Later on, we also expand our research towards protein dynamics, including nascent proteins, protein degradation, protein synthesis, and inhibition, and protein translocation. How protein modifications regulate protein translocation. Next, including how protein glycation can impact protein dynamics. Besides, we are also working on some biomedical applications. Here, I'd like to highlight one direction we have been working on. That's the surface glycoprotein study. Next. So as you know, cell surface contains many important proteins, from transporters, receptors, enzymes, binding proteins. Pretty much all surface proteins are glycosylated. As you may know, cells, either mammalian cells, yeast, bacteria, they are covered with glycans. These glycans are bound either to lipids or to proteins. In order to characterize the glycoproteins only on the cell surface, we have to selectively separate them before mass spec analysis. Otherwise, mass spec can tell these proteins from surface or from inside of cells or other places. We integrated metabolic labeling, clear chemistry, enzymatic reaction, and mass spec based on proteomics to specifically and also globally characterize glycoproteins only on the cell surface. I couldn't go more details about that, but yeah, we can do it. Next. And we have developed different methods to target the surface glycoproteins. Some of them already published. And because the method we developed is very mild, this can allow us to study the surface glycoprotein dynamics, including degradation and also dynamics of surface glycoproteins during cell immune response, and surface glycoprotein trafficking and surface glycoprotein interactions. Next, what we can do is we can comprehensively and quantitatively analyze proteins and protein modification. For protein modification, we even can do it science specifically. And what I need help with is we want to apply our method to neuroscience research. Thank you all.
Thank you so much. Our next speaker is from the West Coast currently, Tammy Tran, who is currently wrapping up at Stanford in order to join Georgia Tech School of Psychology in August. She investigates the neural mechanisms of memory encoding and how these processes evolve with aging and Alzheimer's disease. Her research employs virtual reality navigation, neuroimaging, and biomarker analysis to explore how spatial context, cognitive factors, and biological variables contribute to memory variability across the lifespan. She's an avid plant collector and apparently a very good Tetris player, which we are going to judge when she arrives in Atlanta. Thanks for being here, Tammy.
And I just want to clarify, I said Tetris, not tennis. It's Tetris, okay? No, I think that Constance said it right. I just wanted to make sure that everyone knows that I am not that athletic. All right, so thank you so much for letting me be here. It's so great to learn about this community of fantastic researchers doing really interesting science before I've even arrived. And so, as Tansi said, I'm an incoming assistant professor, and my research focus is predominantly on cognitive aging and preclinical Alzheimer's disease. And so I have this learning and memory background, and my research during my postdoc and PhD really focused on understanding what are the mechanisms underlying memory and what could explain memory variability as you're getting older, as well as in Alzheimer's disease. And so I've typically worked and collected data on large cohort studies, as well as longitudinal studies in collaboration with the Stanford Alzheimer's Disease Research Center, as well as the Johns Hopkins ADRC as well. And so beyond just a general interest in aging and AD, what I also didn't mention is that I did my PhD with an animal researcher, Michaela Gallagher. And at that time, I was really focused on translational research spanning between models of aging in rodents and human neuroimaging with Arnold Walker. Okay, next slide, please. Okay, and so my research focus really explores understanding the mechanisms of memory in healthy young adults, and then trying to understand how might they change in aging and Alzheimer's disease. So here I predominantly focus on developing very sensitive memory paradigms. So these are some of the examples of memory paradigms that we use to look at changes in specific memory functions. I'll really have focused predominantly on memory, but I also have looked at like the interaction between like sustained attention and memory and working memory and episodic memory as well. And so my bread and butter are using functional and structural MRI tasks to examine memory function, specifically using univariate and multivariate pattern analyses. I'm also really interested in overall structural changes contributing to memory. So looking at both 3T and 7T. So I recently developed a new deep learning model to identify hippocampal sublayer thickness in 7T, to look at the relationship and see how these sublayer changes might be predictive of the earliest changes of Alzheimer's disease pathology. And as in my postdoc, as this work got more popular, I've also incorporated the use of Alzheimer's disease biomarkers and genetics in predicting memory variability. And so this could include plasma and CSF proteins, looking at like inflammation, looking at just overall glucose, and looking, of course, at tau and A beta 4240. And I think that, you know, one of the things that is really interesting is that not only can this explain memory variability, but can also explain things like cognitive resilience. Why do some people decline? Why do some people not decline? And it can also explain better, it can also control a little bit better and explain more variability, making our relationship between structure and function a little bit more clean and precise as well. The next slide, please. And so could you press play on this video? I don't know if it's going to work. Okay, great. Thank you so much. And so, you know, really inspired by some of Scott's work, where Scott talked about, you know, these individual differences in performance and aging, and how you can use spatial navigation. To look at that, I've also tried to incorporate the usage of immersive virtual reality and spatial navigation to explore, you know, how does space affect memory performance? And I'm very interested in the usage of these real world variables and how they might contribute to things like memory. You know, how does the space that we're in contribute to memory? And more recently, I've also dabbled with the idea of doing longitudinal remote cognitive testing through like applications. You know, I've developed iPad apps in lab to try to understand memory testing and online testing in young and older adults. Sorry, I'll finish up really quickly. Next slide. And so, you know, what I really need help with is I've done a lot of biofluid biomarker processing, but it's always been a relationship with like a neurologist or behavioral neurologist to analyze this data. And of course, if anyone's interested in additional biomarkers of interest, but in my time, I've always worked on large collaborative studies using large cohorts of clinically unimpaired older adults or patient populations. And so if anyone is interested in developing that, I'm super excited. And it's a big goal. Thank you so much.
Thank you, Tammy. Well, we are almost at the end. And the last talk is going to be by Anne Farney from the Georgia IP Alliance.
Hi, good morning. Thank you very much for the opportunity. I'm probably one of the only non-faculty presenter, but I felt like that's my unique contribution possibly to this panel is maybe providing that industrial to academic connection. So personally, I do intellectual property for living, meaning I deliver people's brainchild, like, you know, your brainchild, end product of your brainchild is a grant, and then you submit it and publish paper. That's the process. So in my line of work, I deliver people's invention as, you know, brainchild. So the end product would be a patent, intellectual property, and then that gets industrialized and commercialized. So I have really intense interest in the application of AI as a parent, like as well as in some of my industrial collaborations with folks that are actually doing therapeutic work. You know, they actually set up clinics around the country that are actually treating gambling and substances abuse, addictions, as well as major depressive disorder, possibly even OCD. And as a parent, like I'm just amazed by the amount, because like you can even tell from today's representation, a lot of research is on Alzheimer's. But what about the cognitive behavior of young kids? I felt like we're kind of like missing. Anna probably touched earlier on that, but that seems to be an area that, you know, I don't feel like as a parent, I have 3 kids, I don't feel like I've gotten a lot of support in that area. But there's a lot of vast amount of literature that's out there. People already develop protocols, but how do you use AI maybe to help to implement that, to, you know, to guide the parents as well as the kids to make it a more rewarding experience. So that's another area that I'm interested in. So obviously this can be translated into other areas as well, right? So in terms of maybe just habit formation, as well as breaking bad habits, forming good habits. And then there's, you know, the marital aspect of it, which I'll lightly touch on in a little bit. So second slides, please. So this is I was really delighted to see that TMS was mentioned earlier in one of the speaker's slides. So there are clinics that's set up throughout the country that has actually used a targeted TMS to treat major depression, and the remission rate is more than is less than 60%. It's less than 60%. So it has a really nice therapeutic profile. So what I felt like the cognitive behavioral element could come in would be maybe some of the measurements that some of the speakers has mentioned earlier, some of the biomarkers, or maybe even using the functional MRI imaging to track the development, how to improve that outcome. Maybe so if there's some follow through, like an AI assistant follow through for that kind of treatment, it would help to, you know, to improve the rate of recovery as well as maintaining it. And one of the speakers talk about the microbials that they're developing supplement, you know. So what I'm probably interested in is how do you convert that research, maybe package it up to actually deliver to the patients, and then after they finish their TMS treatment, I hand them a bottle they can take home. Okay, next slides, please. So this is, I've already mentioned it. So this is one of the things I was thinking about is, like, for example, Smart and Scattered talks about a lot of the cognitive behavior milestones, but how do you help kids to accomplish that? Similarly, you know, Atomic Habits, and these are all along the same lines. So next slide, please. So what I can offer, obviously, I can offer possibly connecting some of the researchers to get connected with some of these clinical data, further analyzing it. And then if there's any commercial interest in any of your research, it may be converted, you know. So that's where I could come in. What I need help with is I'm really grateful for this opportunity that I get to know all these fascinating research that's out there, and I can see, you know, if I can incorporate some of that into my some of my commercial development, you know, that'd be pretty awesome. Thank you very much.
Thank you very much. Well, I just checked, Yurt Alumni is not here, so we are going to have 13 talks rather than 14. That also means that we have about 16 minutes for Q&A and brainstorming session. Let's get started with a big applause to the entire set of our speakers, whose talks have been amazing, showing us that in our environment over here in the greater Atlanta region, we have everything from chemical tools all the way up until the intervention with the human brain. It's an exciting place to be. Now, before we get started with the open round brainstorming session, if there are any questions to any of the speakers, please unmute yourself, and then we can go through those questions first. Maybe I'm gonna get the ball rolling. Ratan, in one of your slides, you mentioned that in the current biology paper, recent paper, you guys found the region for food in the human brain.
Yeah.
So I wonder how universal is that?
Oh yeah, so happy to tell you about that. So well, an open question is, like why do some specific categories have modules in different parts of the cortex, right? Like in visual cortex, we know there's something about faces. Now the question always has been like, what other categories exist for which we have modules, right? So what we did in our study, I'll tell you that first, we analyzed a very large data set. And what we found was if you do some kind of component modeling, so it's something like an ICA. It's like a Bayesian version of the ICA. It's called non-negative matrix factorization. You can extract specific modules for the parts of the brain that we know, things like faces and scenes and so on. And then we found a new thing. And as far as we can tell, that new thing corresponds to images of food. Now we tried many aspects here. We like, what is it about food? Is it a texture of food? Is it color of food? And we tried to rule a bunch of these things out, and we could not. So as far as we can tell, there is a module in every single one of us. It lands in exactly the same location, which is part of your temporal lobe. And every time you look at something tasty or anything that you think is food, it seems to have a pretty high response. Now, the jury is still out there. We know of people who are now recording from monkey brains, trying to see, what is it about food. We are now trying to run experiments ourselves where you take something that, I don't know if you've seen that Netflix show, Is It Cake? Right? Like there'll be like everyday objects like a cup, and then suddenly there'll be somebody who'll like take a knife and just run it through and like, oh, it's a cake. Right. And so we are trying to run experiments like that with both humans and monkeys, where you suddenly show them that it's food, and we basically want to decouple the knowledge of something being food versus the physical attributes of food. And that's one way to do. I think I've taken too much time, but that's our latest on food. Right around lunchtime. I don't know if that was a good idea.
Yes, priming people. Yeah, pretty cool. Are there any questions to our speakers? While you are thinking, if you don't mind, Kathy, would you like to give a little bit of insight to CTSA and the resources that faculty might be reaching out in order to utilize?
Sure, sure. So the CTSA is really here to support clinical research, clinical and translational research. And there are a number of things, as Tansu pointed out, there are a lot of folks here today talked about access to clinical populations. So we don't have a whole series of clinical populations sitting on the shelf, but we do have expertise in recruiting. So there's a recruiting center who can help you set up your study and recruit for your study. We have an informatics section that will, if you need to pull data from the electronic medical record, that will help you set up those searches and help you do the data analytics to get the data that you need. There's a biostatistics core to help you plan your study and then do the right statistics after you collect your data. There are also clinical research centers, and there's one at each institution. So at UGA, Morehouse, and Emory. There's not one at Georgia Tech. But these clinical research centers provide space and expertise. If you do have a patient and you need to bring them in for some sort of study, they will help you set up that visit for a fee. That one's for a fee. So it's subsidized. It's a little bit better. So Lauren just put a brochure in the chat, and that's a summary of all of the things that are offered by the Georgia CTSA, divided by the user. So our website's sometimes a little bit difficult to navigate, but this brochure will help you find things a little bit easier.
Perfect. Thank you so much. Obviously, at Emory and in other hospitals, there are relatively easy ways to access to patient populations, but as you have seen, there's quite a little bit of interest across the fundamental researchers in order to have translational impact also. Are there any questions? Anything that you would like to discuss? We have only 10 minutes for any detailed discussion. Probably it's not going to be enough, but we can definitely get started. And here are some 10 questions that might be worth considering. Oh, apparently there's a word limit, what we can post. And just starting with a few questions that you might be asking yourself in order to see how we can collaborate together and then explore opportunities for funding and research impact.
And we encourage you to reach out to people who spoke today, who might have technique or common interest with you. Also, one outcome of some time of these sorts of events is a group of people get together, want to continue meeting because they share a common interest. They think about maybe putting together a grant, one of the bigger grants. If you do that, we'd like to know about it because it helps to justify why we can go forward with events like this. We will also be sending you a short survey to fill out. Just there you go. Great, put it in the chat already. Just asking for your feedback and whether or not you met someone you would like to collaborate with. So back to the questions. I should have given you some time to read them.
So if I may, maybe I just add like 1 more minute. I totally forgot to mention my thesis advisor was the inventor of Lyrica. It's a small molecule for fibromyalgia. So my PhD was focused on neurotransmitters. So when I saw those synapses, I was like, that's just right in my area.
Wonderful.
They're great questions, by the way, to answer. Thank you for putting those in the chat. Let me throw out one last question to the group. What do you think is sort of the next frontier in your area? What's the really up and coming areas in cognitive neuroscience?
Human brain control, closed loop control, and I'm just jumping, and I'm sure that everybody is going to have a different answer. But at the end of the day, we need to really move from observational science into mechanistic science. The technologies are there, they are not ideal. We still need to go a long way, but the rudimentary technologies that are available right now, they are going to allow us to do closed loop control of the human brain that is going to answer a lot of foundational questions regarding how the brain generates behavior. And I think there's going to be plenty of surprises, given the redundancy in the network, given how the brain operates in a parallel fashion. Inactivating a single region is not going to necessarily give any answer. We're going to need to think about space-time as independent components and start thinking about distributed networks, actually organizing behavior. Therefore, intervening with distributed networks, both in space and time, is going to be required. But at the end of the day, we are going to finally know how the brain generates behavior.
So, I mean, that's what actually got me really interested, and I felt like I didn't really express my ideas really clearly. So that TMS, for example, that's a thing you can touch, then actually produce an outcome, right? But in the traditional psychology, cognitive psychology, that's not part of that. So I feel like we're really in that era, and today when I saw all those presentations, it confirmed what I was suspecting, is that there is a lot of the stuff that we're doing, the researchers are doing, are physical, like the biomarkers you're measuring, the signals you're measuring are physical. So if you think about the classical psychology, like Dr. Like the lobster effect, like if you hold your posture a certain way, that will activate. But how do you measure that? So that I'm really interested. I felt like that's just the beginning. I'm really hoping this would explode, because I felt like that's really the real impact, the real clinical outcome would come from but that lobster effect, maybe I should have put a lobster on my slide, but that's really what I was looking for. And I was thinking, okay, well, if there's some measurable change we could do, like the posture, positive mental imaging, like how do we measure that? And then hopefully that will expedite that therapeutic process.
Yeah, absolutely.
I think in my field, what is becoming more prevalent is more precision-based neuroscience and psychiatry in terms of neuroimaging. So instead of global or averaging across brains, we make maps of individuals, right? So
there we go. Lead. Let's lead meeting.
Sorry, would you, there you go. Go ahead, Ashley, sorry.
Oh, no, it's okay. Yeah, so a lot of my collaborators at WashU are currently working on that, specifically with neonatal brains, but I'm thinking even as far as larger base studies like the adolescent brain and cognitive development study, which has 11,000 participants, but actually mapping brain function and structure by the individual as opposed to these global metrics that I think can lend a lot more to more translational research into psychiatry.
Yes. Yeah. Single trial resolution, single, individual brains, that would be amazing. Also, we need to stop averaging across trials, not only across brains, but also across trials. Yeah.
I mean, like the traditional diagnosis, and even nowadays, the diagnosis is still behavioral based. Like, I'd be maybe we'll get to a point where it would be actually, it's like a blood work you could do. There's some kind of equivalent. You can get a little bit more measurable instead of just behavioral based.
Maybe there are some biochemical, I'm so sorry.
No, go ahead. Go ahead, finish up.
No, go on. No, what I was going to say, maybe there are some biochemical markers, and Professor Wu's research might be able to help.
Yeah, so we're seeing a couple of things show up in the chat, but we really do need to wrap up. I just want to really thank Tansu for leading us today. You did a great job as moderator. I hope all of you heard something that was of interest to you and maybe somebody you'd like to reach out to. Again, please fill out the survey for us, and thank you for coming. You'll have all the contact information in the calendar invite. So thanks again.
Thanks again for organizing this. It has been so much fun, and I'm sure that there are going to be many opportunities. Many discussions are already starting, and we will continue. Have a wonderful day all.
Thank you.